Hi, my name is Vivian Nguyen, and I'm an undergraduate at the University of Arizona. Today, my project is Gene Regulatory and Enrichment Analysis identifies molecular mechanisms of early-onset APC mutation-negative colorectal cancer. To start off with, colorectal cancer is an aggressive tumor type with diverse biological outcomes and patient survival rates. It most commonly is identified with mutations on the tumor suppressor gene APC. Loss of APC function is directly linked to tumor initiation because it results in uncontrolled cell division via the deregulation of the Wnt and beta-catenin signaling pathway. This links to chromosomal instability and a high mutation rate that promote tumor progression. Therefore, biologists have specifically targeted this molecular mechanism in order to direct therapeutic options towards the restoration of the Wnt signaling pathway. However, in 2018, a subtype of colorectal cancer was identified marked by the absence of APC mutations. Our goal for today is to understand the biological mechanisms behind this cancer subtype in order to target potential therapies towards this cancer type, understand the heterogeneity of colorectal cancer, and combat health disparities. To begin with, we identified 84 APC-negative primary tumors from TCGA and matched the APC-positive samples for age and sex. Our original GO-term analysis showed an excessive immune signaling upregulated in the non-APC group. This blocked out more specific mechanistic and biological pathways that we can specifically target on a molecular level. Therefore, we performed an immune correction by increasing the immune signaling from the APC-positive samples. We used an application called CyberSort, which is a digital cytometry application that is able to rank the samples based on an absolute immune score using their gene expression profile. We then optimized the immune infiltration of the APC samples in order to minimize the absolute mean between their immune scores and maximize the p-values. This made the two groups statistically indistinguishable. In order to validate this correction, we compared the slide lymphocyte percentage before and after the adjustment. And as you can see before the adjustment, there was a statistically significant difference between the slide lymphocyte percentage. However, after we optimized the immune infiltration in the APC group, there was no longer a statistically significant difference between the two groups. From here, we performed gene set enrichment analysis between the non-APC mutated cohort and the APC mutated cohort. As you can see, there's a strong metabolic signature upregulated in the non-APC group, as well as DNA repair signaling. This suggests that the mechanism for cancer development in the non-APC group is involved with the reverse Warburg effect, where cancer cells act more stem cell-like and prefer oxidative phosphorylation as opposed to anaerobic respiration. However, on the other hand, we get a lot more specific signaling from the APC mutated group. This is due to the fact that APC encodes a multifunctional protein involved in several processes, including signal transduction, cell adhesion, migration, proliferation, apoptosis, and cell differentiation. In order to better understand where this metabolic phenotype is coming from, we performed cluster analysis on the differentially expressed genes between the non-APC and the APC mutated groups. On the top, we have the genes upregulated in the non-APC group. And as you can see, the gene expression is clustered into 6 different groups, which we performed GO-term analysis on each group. However, our GO-term analysis only picked up general ion transport and regulation terms. However, on the other hand, the downregulated groups or the upregulated genes in the APC group have a more consistent gene expression signature. From here, we perform GSEA on the transcription factor target differences between the two groups to figure out which transcription factors are regulating this metabolic phenotype. Here we list the top 20 upregulated transcription factor targets in the non-APC cohort. We compared our results to methylation analysis using MEthylMix, which identifies differentially methylated regions associated with gene expression. The mean column over here represents the mean difference between the APC mutated samples minus the non-APC mutated samples. A positive mean shows the genes that are associated with hypermethylated regions in the non-APC group, whereas a negative shows the hypomethylated regions. Most consistently between these two analyses, we see the homeobox or the HOX familial transcription factors show up in both of these analyses, showing that hypomethylated homeobox genes might be a biological marker for non-APC mutated colorectal cancers. In conclusion, we plan to look further into HOX, as it's been previously linked to cancer malignancy because of its role in embryonic development. Other potential candidates we can focus on are PRAX and PRAX2 genes that are hypomethylated in the APC mutation negative cohort, as mutations in these genes have previously shown up in colorectal and prostate cancers. Overall, our future research will be targeted towards being able to understand the mechanism for cancer development that is causing for this metabolic signature. That will be it for my presentation today. Thank you for watching.